A skeletal muscle consists of muscle fibers. Each muscle fiber, or muscle cell, extends the length of the entire muscle. 10 to 100 muscle fibers are bundled together by connective tissue into fascicles. Myofibrils are present inside each muscle fiber. Myofibrils consist of thick filaments and thin filaments. Each myofibril is made up of a series of contractile units called sarcomeres. Z-discs connect adjacent sarcomeres. The thick myofilaments are composed of bundles of myosin molecules, each shaped like golf clubs that have been twisted together. The thin myofilaments, which are anchored to the Z-discs, are composed of bead-like molecules of actin that are twisted around each other, with binding sites for myosin. When the muscle fiber is at rest, these binding sites are covered by tropomyosin molecules that are held in place by troponin molecules. The sliding filament model describes how skeletal muscles contract. Skeletal muscles stay at rest until stimulated by a nerve impulse traveling down a neuron to the muscle fiber. The nerve impulse triggers the release of calcium from the sarcoplasmic reticulum, the muscle cell's specialized endoplasmic reticulum. Calcium binds to troponin on actin, causing tropomyosin to shift and uncover the binding sites. The myosin heads, activated by ATP, bind to actin, forming cross bridges. The myosin heads pull the actin toward the center of the sarcomere. Each myosin head then detaches and, using energy from ATP, then attaches at the next binding site on the actin. This pulls the actin toward the center of the sarcomere. This process continues, with the thin filament sliding over the thick filament. The thin filament sliding toward the center of each sarcomere quickly shortens the entire myofibril, and thus the entire muscle fiber. This diagram illustrates the sarcomere in a relaxed state, then an intermediate contracting state, then a fully contracted state.